24,000. You see the polyploid endosperm. Endosperm formation is taking place by the double petalization in angiosperm, whereas in the gymnosperm, it is forming before the petalization by division of female gamete, applied female gamete, directly applied female gamete, directly taking the division and it forming the endosperm. That's why in the gymnosperm it is applied, and whereas in the angiosperm it is diploid. Why? The second male gamete fusing with the second male gamete fusing with the diploid polar nuclei. That's why three nuclear fusing taking place. That's why we're calling it as triple fission. Endosperm formation is taking place in angiosperm by which process? The completed exam they asked. Which process it is? Triple fission in the double petalization. In angiosperm, double petalization is taking place. Double petalization. Okay, that's first petalization, first male gamete fusing with the male gamete, first male gamete fusing with the egg cell, and second male gamete fusing with the polar nucleus. Two times fertilization taking place. That's why double fertilization. Here the three nucleus are fusing. That's why calling it as triple fusion. By the triple fission, pen primary endosperm nucleus will be formed. That primary endosperm nucleus will become the endosperm. Endosperm is a nutritious layer, nutritious membrane, the nutritious cells that supply the nutrition to the development of the embryo for the embryo maturation, embryo development, and also for the survivability of the embryo. Okay, that is the reason for the formation of endosperm and fluidity of the endosperm. That is the reason in the gymnosperm, endosperm is applied, whereas in the majority of the angiosperm, endosperm is diploid. Common type of the embryo sac is polygonum type. In the 7 cells, 8 nucleus are there. Of the 2 nucleus are polar that fuse with the secondary male gamete, forming the triploid nucleus. They are also there in the allium, drusa, adaxa type. We have studied embryo sac types. These are the types of the embryo sac. Polygonum and Enothera type is mono and diploid, allium and Enothera is a diploid, and these all remaining are tetraploid type. What are those? Peperomia, Pinia, Drusa, Adaxa, Fritillaria, Plumbagella, Plumbago. These all are 4 nucleus. That means the 4 nucleus after reduction division, for the four nucleus will be remain. It will be participate in the development of embryo sac. But is only one nuclei is participating, that is monosporic type. If the 2 nuclei is participating, that is disporic type. If the 4 nuclei are participating in the embryo sac development, that is tetrasporic type. These 2 are the monosporic type. These 2 are the disporic type, Allium and Endomia, and the remaining Pinia, Fritillaria, Drusa, Plumbagella, Plumbago, Adaxa. These are 4 tetrasporic type of the endosperm. That means 4 nuclei will be taking place in the development. And you see in the Pinia, Fritillaria, Plumbagella, Plumbago, there is a 5N stage of fluidity. There is a 5N stage of fluidity is there in the Pinia, Fritillaria, Plumbago, and Plumbagella. Why? There is a 4 nuclear stage of the polar nucleus. That's why there is pentaploidy type. That is pentaploidy type. Okay, that is the reason. Okay, here you can see the 4 nuclei at the middle. Here also 4 nuclei, 4 nuclei, polar nucleus, 4 nuclear, polar nucleus. That's why 4 plus 1 second male gamete. So that it forms the 5N endosperm, 5 nuclear stage. Whereas in the peperomia, the middle 8 nucleus are there at the middle. 8 nucleus plus 1 male gamete. 8 plus 1. One male gamete. So that 9N stage endosperm is forming in peperomia. So many in the competitive exam is asking. Whereas in the RM, highest polyploidy type of the endosperm, that is 24,576 nuclear stage. These many number of the nucleus are present in the RM type. That is a variation, that is the formation of the endosperm. Variation in the endosperm because of the polar nucleus and because of the formation of the endosperm before the fertilization or after the fertilization. Here you can see in the gymnosperm, endosperm is forming before the fertilization. Here you see the female gamete, all the female gamete is taking the division and it forming the taking the division and it forming the endosperm. And it forming the endosperm. You can see here the endosperm. All the female gamete will become the endosperm before the fertilization, whereas the egg cell will become the embryo. That is there in the gymnosperm. But in angiosperm, you can see the what I told you, the polar nucleus, that means central cell nucleus, 2N stage, fusing with the second male gamete, so that it is forming the endosperm layer. You see the endosperm layer, endosperm, endosperm formation after fertilization by the triple fusion. You see endosperm layer in the monocot and in the diocot of the angiosperm. Okay, that's why it is triploid in angiosperm, haploid in the gymnosperm. That is the reason. Okay, and then you can see there is no endosperm in the Orchidaceae and Podostomaceae, Trapeaceae. That is very, very important. Not endospermic. Endosperm, there is no endosperm formation. In which family endosperm formation is not taken place? There is a question. Orchidaceae, Podostomaceae, Trapeaceae. Non-endospermic, no endosperm in the seed. Only embryo is present. No endosperm. Okay. And then endosperm again, two types based upon the utilization of the endosperm. If the all the endosperm will be utilized, if all the endosperm is completely utilized, no endosperm in the seed, only embryo cotyledons is present, no endosperm if it is present, that we call it as non-endospermic or exalbuminous seeds. Albumin, endosperm. Albumin is totally utilized by the embryo for their development in the seed. That is non-endospermic. That usually they are in the pulses. In the pulses, especially in the Leguminaceae, Fabaceae, Sessalmaceae, Mimosaceae. There is no endosperm, only cotyledons will be present. The fully loaded cotyledons are present. That's why the that's why the pulses endosperm is totally utilized. Whereas if the endosperm is remain in the seed, even after utilization of the endosperm for the development of mature of embryo, some part of the endosperm will be remain. That endosperm we calling it as perisperm. What we calling it as perisperm. This perisperm, if it is present, that we calling it as endospermic seeds or albuminous seed. If the endosperm is remain even after mature of the embryo, even after mature of the embryo, if the endosperm remain, endosperm is present, that is endospermic seeds. Paddy, wheat, maize, coconut, castor oil, castor bean. In this all, the endosperm is present. That all endospermic seeds are albuminous seeds. And non-endospermic seeds, if the total endosperm is being utilized. Okay. And then on the basis of their development, on the basis of utilization, two types. Okay, albuminous endospermic and non-endospermic. On the basis of development, there is a four type. How many types? Four types of the endosperms. How many types? Four types of the endosperms are there. That is nuclear endosperm. There is filled with the nucleus. You see the all the nucleus are there. There is no covering sheath, cell wall. Only the nucleus are present in the embryo sac. In the embryo sac, all the nuclear endosperm nucleus are present. That is nuclear endosperm. And the second one is cellular endosperm. Simultaneously, whenever the endosperm nucleus is taking the division, immediate after nuclear division, it also taking the cell division. So that each one will become the each cell, separated cell. So that all the embryo sac is filling with the endosperm cells. Endosperm cells, each cell with the one nucleus. Endosperm cells, cellular. It is filled with the cells. And then the third one is helobial. That means the both the type. Cellular type of the endosperm at the chelazal end and nucellular type of the endosperm at the micropylar end near to the embryo. And the both the type of the development is there that we calling it as helobial endosperm. That we calling it as helobial endosperm. And then the next is ruminate endosperm. After formation of the endosperm, at the maturation of the endosperm, it will be folded. It forms uneven shape endosperm. That is ruminate endosperm. And also aileron layer, also the outer layer of the endosperm, that also we will discuss. These are the types of the endosperm based upon the development. And based upon the utilization, two types, endospermic and non-endospermic. Okay, these are very, very important. And then we will go each and every one. You see here clearly in the division, whereas in the cellular endosperm, immediately, simultaneously, after the nuclear division, cell division also taking place. One cell, one nucleus divided into two. So that cells also divide into, embryo sac also divide into two. And then 4, 8, 16. Like that, immediately after nuclear division, cell division is taking place. Simultaneous division. cell division with the nuclear division, so that it is forming the cellular endosperm. Here you see the embryo, the remaining part is the endosperm. And whereas in the nuclear endosperm, only nuclear division is taking place, no cellular division, though no cells formation is taking place. One nucleus are justified, nucleus are fully loaded in the embryo sac. It formed layers of the endosperm, outer layer, inner layer, like that it formed the endospermic layer. Endospermic layers it's formed. And the whereas helobial endosperm, in the first division, the two nucleus, micropylar side nucleus division is not taking place, cell division immediately after nuclear division. Whereas the micropylar chalagal end nuclei division, immediately after nuclear division, even cells also taking the division. So that it is forming the both the type, cellular and nuclear. Cellular and nuclear. Both the type of the endosperm is there in the single embryo sac. That we are calling it as helobial endosperm. What it is? Helobial endosperm. This is the developmental stages in the endosperm. Cellular endosperm, nuclear endosperm, helobial endosperm. You see here, whenever the primary endosperm nucleus form, that primary endosperm nucleus is taking the mitotic division, so that it is dividing into multi-number of the nucleus, multi-number of the nucleus. Okay, here without the cytokinesis, only karyokinesis is taking place. Nuclear division is taking place. No cellular division is taking place. So that all the nucleus are spreading in the embryo sac. There is a more number of the pre-nuclei in the cell. That's why that we are calling it as nuclear endosperm. You see, usually at the chalagal end, more nuclei are present than the micropylar end. Okay, sometimes later cell wall formation will take place periphery toward the center. That is especially in the maize, rice, wheat, cotton, and sunflower. The multicellular endosperm is formed. That endosperm, you see in the Acalipa indica, how the endosperm formation is taking place. Nuclear endosperm is forming, taking place. Even in the coconut, cell wall formation is incomplete. That's why the outer multicellular solid endosperm and inner multinucleated, that means pre-nuclei liquid endosperm. The coconut milk. Coconut, sometimes the outer layer is taking the cellular division, so that it forming the coconut, kobbari. Whereas inside, at the middle, there is no cellular division, only nuclear division is taking place. That's why that is liquid pre-nuclei, liquid endosperm. That liquid endosperm, we drink the coconut water, and even solid endosperm also we eat as a coconut. We extract the coconut oil from that. You know, that coconut, what we are eating is endosperm. Okay. But in the groundnut, it is not the endosperm, the cotyledons, from the cotyledons. Okay. Whereas in the maize, white part is there now below the seed, that is the endosperm. Remaining all the maize is yellow in color. But whereas at the bottom, the white color is there, that one is the endosperm. You can see here, not the endosperm, embryo. Here the remaining all the yellow part is endosperm, all the yellow part, whereas the white part is the embryo. You can you can distinguish that easily in the image. Easily you can see the cotyledons in the pulses. Easily you can see the uh, endosperm in the grasses, especially in the wheat, paddy, maize. Okay, you see the liquid sinusidium, it forms the coconut meat that we eat, coconut. Okay, and then you can see the various type of the endosperm hostorias. What happened? Sometimes the cellularization of endosperm is restricted at the micropylar end of the embryo sac. For the development of the embryo, the cellularization of the endosperm will be stopped. That time, what happened? The endosperm will be formed extra appendages to absorb supply the endos to supply the nutrition to the embryo for the development. That time, it formed the special hostorias to supply the nutrition. Whereas the hostorias in fungi to absorb the nutrition from the host. Like here also, it is forming the projections to absorb, to supply the nutrition to the developing embryo. That are hostorias. You see in the crotalaria and the grevillea and the lomancia, hostorias are forming by the endospermic cells. That's why endosperm hostoria for the supplying of the nutrition to the developing or growing embryo. That may be tubular hostorias or finger-like projection hostorias, various types, which are there. Usually tubular hostorias are there in the Cucurbidaceae, Fabaceae, and Proteaceae family. And there is, you can see the hostorias. You see the scleria endosperm hostoria, how it is developing at the microphylar end, and sometimes at the chelagel end also hostorias are formed. Okay, and then you can see here the cellular endosperm. It is opposite to the nuclear endosperm. What happened here? There is no here immediately after the nuclear division, mitotic division of the endosperm, it takes the cellular division also, so that it forms the multicellular structure endosperm. Multicellular structure endosperm. Okay, no pre-nuclear stages at any stage. So after that, even after that, whatever the cellular endosperm, it also further divide and it form 2, 3 layers of the endosperm. Okay, you see the cellular division of the endosperm is forming the for the coconut milk, and petunia, datura have this kind of the endosperm, that is cellular endosperm. Okay, you see in the drainage how the cellular endosperm fully covered with the cellular endosperm cells. Here you can see various cellular endosperm type hostorias. Especially these all are forming in the microphylar end. You can see that the chalagelan, at the chalagelan, these all are forming special projection structures Iodina rhombifolia. You can see the long projection of the Astoria to absorb and to supply the more nutrition to the developing embryo. Okay, you can see here the endosperm, how long it is projected. The endosperm, it is projected. You see here the endosperm. You see here the endosperm, how much widespread and the absorbing the nutrition and supplying the nutrition to the embryo, developing embryo. In the Borrelia cristata, how it is the endosperm Asturias are forming like this. And then a helobial endosperm. There is an intermediary. That means half of the cells, that means at the chalagal end, immediately after the nuclear division, cell division is taking place. Whereas in the micropylar and free nuclear space is there. That means the both types, pre-nuclear endosperm cells and cellular structure of the endosperms. Both the types are there. That's why it is forming the both intermediate type of the endosperm that is called as helobial endosperm. And you can see the micropylar chamber regular wall formation is taking place, but in the chalagal end, the wall formation is not taking place. In this, Hastoria develop from the micropylar tissues. Tubular structures, outgrowth, it penetrates into the new cells at the chalagal end. It penetrates into the new cells at the chalagal end, and then it absorbs the nutrition from the new cells. That means endothelium, nutritious layer, new cells. It absorbs nutrition from the new cells supplied to the embryo. Okay. And then you can see the various type of the helobial endosperm and Aspergillus tenuifolium. You can see how the Hastorias, how the helobial endosperm formation is taking place. The both cellular and nuclear. And the fourth one, the endosperm is ruminate endosperm. What type it is? Ruminate endosperm. That means irregular, uneven surface. Irregular and uneven shape surface, not the smooth. Folded. If the endosperm is folded inside irregularly, uneven shape, that type of the endosperm is called as ruminate endosperm. That is usually there in ananas, komoda, sitapal, and Meristica fragrans, Georgica, Japatri, and Aralaceae and Araceae. Araceae. Araceae, you know the Areca catachu, Pokavaka. Areca catachu, Pokavaka also, it is ruminate endosperm. Folded endosperm. Endosperm is tightly folded, very hard endosperm. Even in the Georgica, Japatri, Meristica fragrans also, endosperm is tightly folded, very tough. Georgica, Japatri. You see in the Japatri, Meristica, folded endosperm. It folds inside and it will be very tough, very hard. It will be reflect the activity of endosperm as well as seed core. By folding the very tough, rough type of the endosperm formation take place, and also activity for the main to observe to preserve the more nutrition in the pores. The both the ways. And the mosaic endosperm. That means the not one type specific type of the endosperm. Okay, there is no uniformity. Some of the endosperm tissues are yellow, and some of them are white color in different color, yellow and white. You see mosaic pattern of the endosperm tissues. Due to failure of triple fission, our male gamete may not fuse with the polar nuclei. That time, male gamete is forming one type of the endosperm and polar nucleus forming the other color type of the endosperm. So the both the type of the endosperm formation is taking place. Or otherwise, the male gamete and the polar nuclei may taking the division separately, independently, so that it forms the two distinct color nuclear endosperm will be formed. So that at the maturity, endosperm will be giving the mosaic type, variegated appearance. In the image, you can see clearly yellow color and white color. At the bottom, white color is the embryo, and the inside, the lower side, white one and yellow one. This is the endosperm. Mosaic, two types of the endosperm. That's why it is a mosaic endosperm. This is there in the mage, Gia mage and Gia mage. And one more important is aileron layer. It is very, very important for every seed. Aileron layer is a very, very important in the endosperm. It is very useful for the seedling, for the germination of the seed. Endosperm layer, endospermic aileron layer, outer layer of the endosperm. The outer layer of the endosperm, we calling it as aileron layer. Outer layer. The outer layer of the endosperm, allocation color with rich proteinaceous. This rich proteinaceous layer, it will be useful for the stimulation, for the activation of the enzymes, especially it having the amylase enzyme, which having the amylase. This amylase enzyme, it activate the, what it do? Amylase enzyme, which break the starch and it separate into CO2 and H2O. That water molecule, whenever it is observed, then gibberellic acid, which is there in the embryo, will be activated. Whenever the amylase it is activated, it forms, it releasing the energy and water molecule. That energy and water molecule will be activate. Energy and water molecule, it is activate the enzymatic activity in the seed for the germination. Then it starts gathering of the energy. With this energy, they stimulate the gibberellic acid, the amylase enzyme is activated gibberellic acid. So the gibberellic acid is, it is the gibberellic acid, one of the phytohormone, it activates, it motivates, it stimulates the germination of the seed. To control the seed dormancy, to stop the seed dormancy, we use the gibberellic acid. For that reason, the aileron layer is very useful for the activation, for the starting of the enzymatic reaction in the seed for the germination. With that, the phosphate reaction, ATP synthesis will take place. That ATP energy will be utilized for the germination of the seed by stimulating the gibberellic acid. Okay, there is a reactions behind the germination of the seed. The amylase, it split the starch molecule into carbon dioxide and water. At the respiration, ATP also released. That stimulation energy will be utilized for the stimulation activation of the gibberellic acid so that it starts the germination of the embryo. So that's why the water is needed for the germination of the embryo seed. What are the functions of the endosperm? So more functions are there. The very rich source of the proteins, carbohydrates, and fat, which are very useful for the seedling and for the seed germination and the maturation of the seed and for the development of the embryo. The endosperm is very, very useful. At the time of fertilization, only few less amount of the nutrition available in the embryo sac. But with the formation of the endosperm, this endosperm division is taking place. This endosperm is having the enough source of the food, so that it endosperm, it supply the nutrition to the embryo for the maturation, for the embryo formation, for the development of the embryo, and also up to the seedling of the seed. You see, the coconut milk and zeatin from the maize that we are using as a nutrient medium for the in vitro embryo culture, for the culturing of the embryo, for the development of the embryo, for the germination of the embryo, seed embryo. Like that, there is a lot of usage out there in the endosperm. Even coconut milk we use in the tissue culture also as a nutrient medium, nutrient supply, if the other media are not present. Even in the coconut milk also, we can grow the callus. Okay. Endosperm, it provides a nutrient to the seed during the dormancy and stimulate the seed during the germination, seedling. It also contains the hormone cytokinin and gibberellin. It helps in the cell division, cell differentiation, and gibberellin in the germination of the seed. In the coconut water is the best example of the liquid endosperm. And in the wheat, corn, maize, paddy, the endosperm is the main source of the food. It shows the regular gene expression and seed germination and include the signals according to the environmental condition, whether the embryo development is needed or not, whether the embryo maturation is needed or not now, whether the embryo germination, seed germination is needed or not. That all will be regulated. The seed germination and gene expression will be regulated by inducing the signal by the endosperm according to environmental condition and the availability of the nutrition. Okay, there is the endosperm, endosperm structure and endosperm types. Okay, endosperm structure, endosperm types, and cellular endosperm, nuclear endosperm, and helobial endosperm, ruminate endosperm, and aileron layer. These all we have studied. Okay. Okay. Have you listened this all? Here you can see the various regions are there in the endosperm development, and various functions also is there in the endosperm. for the endosperm. You can see here for the variation among the species and very much significance of there in agriculture for the endosperm, in the development, in the maturation, in the seedling, in the cellularization. That you see so many reasons for this development of the endosperm, double fertilization, and then sinusoidal stage, that means pre-nuclear stage endosperm, as like in the coconut milk, and cellularization, and as like in the maize, in other ruminant endosperms, and nutrient accumulation is taking place in the endosperm. All the endosperm will be fully loaded with the nutrients so that it will be useful for the developing embryo and also seedling. And it also regulates the genes and hormones. I already told you in the seedling, in the germination of the seed, endosperm is taking part, very precious part in the germination, the activation of the amylase enzyme, and so that activation process of ATP synthesis, and then development of the embryo, development of the seed, and cell composition, and the types of the seed composition, and nutrient provision is there provided by the endosperm, hormonal balance, hormone, cytokinin, gibberellin hormones, and as a food source, in especially in the grains, and liquid endosperm in the coconut, endospermic food in the so many seeds, okay, especially in the grasses, seeds, and aileron layer for the germination and motivation, seedling, embryogenesis purposes, and even in the orchid seeds formation. These all are very, very much useful in the seedling and seed development or embryo formation. Okay, then we will discuss the polyembryony, that we will discuss in tomorrow, I think. Huh? Already time up now. The next class may be start. Huh? The polyembryony means formation of the more embryos. Formation of the more embryos. Usually in the one seed, only one embryo is present. In the one seed, only one embryo is present. But sometimes what happens? The more embryos will form in the single seed. In the single seed, sometimes usually only one embryo is present. But sometimes the more than one embryo, two or three or four or multi-branch embryos formation will take place. Cleavage polyembryony. That type of the embryos, more embryos, okay, within the single seed, that is polyembryony. Polyembryony first reported by Anthony van Leeuwenhoek, the orange seeds, okay, and then word coined by Braun. Polyembryony word first coined by Braun, okay. Usually polyembryony is present in the gymnosperms. Polyembryony is present in the gymnosperms. The common polyembryony is common in the gymnosperms, many gymnosperms. Uh, Rarely, the polyembryony is present in even in dicots and monocots, even in the dicots and monocots. Okay, but usually it is common in the gymnosperms. Okay, shall I continue or shall I stop? You can see here the polyembryony may be taking place, the simple ovule with an embryo sac or multiple ovule with more embryo sac. The ovule with one embryo sac, it may be two types, asexual, gamete without the gamete fusion, gametophytic and sporophytic. whether it is forming from the gametopoietic cells, that means uh, synergids which surrounding the egg cells, or antipodal cells in the embryo sac. Sporophytic cell. Okay, what happened here? Where we stop? Yeah, these are the two types. That means ovule with one embryo, ovule with multiple embryos. Usually, it is happen in the sexual reproduction, either from the zygotic cells or from the synergic cell. It may be polyembryony will take place. Usually, one seed, one embryo is present, but usually polyembryony is normal in the citrus, lemon, and in mango. These two polyembryony is common. The polyembryony basically two types: spontaneous polyembryony and induced polyembryony. Spontaneous means it will be happen naturally by the environmental condition or sunlight, whatever it is the condition. Induced polyembryony means by inducing by special force, external force, or by experimentally, if we induce, if we initiate, that is induced polyembryony. And then you can see the again Ernst divided the spontaneous polyembryony into two categories. Natural type of the polyembryony divided into two, that is true polyembryony and false polyembryony. That means true polyembryony means the two or more embryos rising from the same embryo sac, either from the egg cell or from the synergids or from the antipodal cells or from the nucellus or from the integument. These all are there in the embryo sac only. If these all embryos are forming from the embryo sac cells, egg cell, synergid cells, or antipodal from antipodal cells, or from the nucellus, nutritious layer, diploid nutritious layer, or from the integument, if it may forming the extra embryos, that is true polyembryony. You see, the false polyembryony, that is in the same ovule, but not from the embryonic tissues, other than the embryonic tissues. If other than the embryonic tissues in the ovule, that is false polyembryony. From the placenta, in the Lorantheae, from the same ovule, in the Fragaria. You see polyembryony, true polyembryony, again divided into types are there. Cleavage polyembryony, adventive polyembryony. Cleavage polyembryony and adventive polyembryony. You see, in the polyembryony, I got into genetic basis divided into gametophytic and sporophytic. If it is forming from the gametic cells of the embryo sac, after or without fertilization. Gametic cells means, just now we have seen, the gametic cells means where it is, synergids or antipodal cells. Sporophytic cells means nucella or integument. Okay, either from the synergid or from the integument or antipodal cells, either from synergids or antipodal cells. If it is forming, that is what it is, gametophytic. And if it is rising from the zygote, sporophytic tissues, nucellus or integument within the embryo sac, that is sporophyte. That means from sporophytic tissues. Okay, and then usually Boswinkel divide the polyembryon into 4 types. You see here, as for Weber, the polyembryon is classified into 3, that are cleavage polyembryony, that means embryo Hello? It is live streaming or not? You see simple polyembryonic. You see this type of embryo develop a result of after fertilization. Several archegons, several embryos, several egg cells. Rosette polyembryonic, that is common in the gymnosperms, especially in the pioneers. Additional embryo develop in the rosette cells. You know the while embryo developing in the rosette cells. It form the rosette polyembryonic. You see, angiosperm polyembryonic basically 5 types that are cleavage polyembryonic, embryo from synergids, embryo from antipodals, embryo from endosperm cells, and adventive polyembryonic. These are the 5 types of the polyembryonic which is there in the angiosperm. The first one is cleavage polyembryonic, identified first by Swami in the Orchidaceae family, Halaberry and Eulopia and Cymbidium bicolor. That means proliferation or budding or division of the embryo. One embryo is dividing into 1, 2, 3 like that. The embryo is taking the division, cleavage, splitting into 2, 3 or 4 or 5. Here is the cleaving, splitting. 6 embryos are formed by the proliferation of the suspensor cell that you see here. Either 2 or 3 or 4 is forming by the splitting of the embryo. You see origin of embryo from the synergids. You see, in the embryo sac, egg cell, 2 side, 2 synergids are there. Suppose sometimes even synergids also taking part in the embryo formation along with the egg cell. Synergid cells also male gamete that the fallen tube fuse with the synergids along with the egg cells. That time it formed the more embryos, normal egg cell embryo and synergids embryos. Sometimes with the stimulation, synergids also forming the embryos without the fertilization. Okay, and then in some, what happened? Origin of embryo from antipodal cell. Usually, the antipodal cells, these are there at the chalazalan. These are vegetative cells, but sometimes with the stimulation, these antipodal cells also interfere in the formation of embryos. Okay, you see whether these embryos are attain that nutrient or not yet known, but more embryos formation will take place. Okay, that is happening in this type. Okay, and then you can see here, these are embryos other than the normal egg cell embryo. The antipodal cells also forming the embryo